Hello everyone. Today we're going to be talking about membranes, receptors, and endocytosis. These are listed as two lectures on your schedule, but it's just going to be one combined lecture here. So I think I have 40 or so slides, and at the end I have the study guide for these lectures. An outline of what we're going to be talking about today, just an overview of response and signaling, talk about the different classes of hormones, and then talk about the receptors and the receptor-coupled signaling systems, specifically G protein coupled receptors, ion channel receptors, and then tyrosine kinase linked receptors, and as well as receptors with intrinsic enzymatic activity. And then I'll also talk a little bit about hormone-like lipids, which is relevant to this lecture as well. In general, the cell's survival depends on an intracellular communication networks that coordinate growth, differentiation, and metabolism. Cells adjacent to one another frequently communicate through cell-cell contact, but there's other forms of communication that cover larger distances through extracellular signaling molecules, or hormones. And we're going to talk about how this process works. Here's a diagram from your book. Essentially, you can have things like polypeptide hormones bind to receptors. This can lead to signaling cascades. Let me get my pointer up here. This can lead to signaling cascades. It can ultimately lead to transcription factors that can bind to the DNA and either stop gene expression, slow it down, or make it go faster in some cases. You can have steroid hormones that directly interact with transcription factors. We'll talk about those. Nitric oxide is also can be considered in this process, and I believe I'm going to be talking about that in a separate lecture. But all these things can lead to downstream effects, whether that's secretion of certain proteins, exocytosis, apoptosis, phagocytosis, all through these long-range interaction using signaling hormones. In general, signals are generated and are sensed and are processed by cellular signal transduction cassettes that essentially detect, amplify, and integrate external signals to generate the appropriate cellular response. The three major components are specific cell surface membrane receptors, effector signaling elements, and regulatory proteins. And we're mainly going to talk about the hormones and the receptors in this lecture. The 6 steps in general of cell response and signaling are: first, there's a synthesis of the signaling molecule, that would be the ligand. Then release of the signaling molecule, transport of the signaling molecule to the target cell, binding of the ligand by the specific receptor protein, followed by a change in conformation. Then you get a change in cellular metabolism, function, or development. So this would be the cellular response. And ultimately, you have removal of the ligand, terminating the cellular response, typically through degradation of the ligand. Let's talk about hormones first. So these are chemical messengers, either steroids, amines, peptides, or polypeptides, and we'll talk about those in a minute. These are produced and secreted by endocrine cells into the bloodstream. They regulate, integrate, and control a wide range of physiological features through gene expression and protein synthesis, and we'll get into some details here. In general, there are 2 types, autocrine versus paracrine. Autocrine hormones act on the same cell that secreted them, shown here, whereas a paracrine hormone secreted by one cell and act on neighboring cells, shown here. Pretty simple concept. There are 4 classes of hormone types at a high level. The first one being steroids, the second one amine-derived hormones, and then peptide hormones and polypeptide hormones can kind of get mixed together, but we'll talk about each of these classes. First, the steroid hormones. As we've talked about in the past, these are cholesterol is a precursor to these steroid hormones. And there are three main groups: the corticosteroids, androgens, and estrogens. And these are produced by the endocrine glands, either the gonads and the adrenal cortex. And they immediately diffuse out of endocrine cells into the bloodstream. And because they are lipid soluble, their receptors are located inside target cells. They can move across the membrane of target cells. They are slower acting and have longer half-life than peptide hormones. Again, cholesterol is the parent compound for all steroid hormones, and I've shown you the complex structure of steroids before, or of cholesterol before. In the adrenal cortex, you can make aldosterone or cortisol. You can see these things get a little bit more water soluble than cholesterol. In the ovaries, you produce estradiol, which is just an estrogen. As I mentioned, what happens here, you get hormones diffused through the phospholipid bilayer, so they're either it's a free hormone in the bloodstream or with a transport protein. But in the first step, they diffuse through the lipid bilayer of the target cell. Then in the second step, they bind to the receptor, either turning on or off specific genes. And then the messenger RNA directs synthesis of new proteins, and then the proteins can alter activity. The receptors themselves, the receptors that bind to the steroid hormones, are cytoplasmic receptors, as we mentioned. They are not in the lipid bilayer. They can also transduce signals from other small hydrophobic signaling molecules, such as those listed here. But most of these are transcription factors that bind to regulatory regions of a gene that are responsible to the particular steroid or thyroid hormone. So essentially the hormone binds to the transcription factor, and then that can impact a transcription. What happens is there's a ligand binding, which the steroid binding to the transcription factor induces a conformational change that either activates or represses gene expression. That's steroids. Let's talk about the next class, amine or amino acid derived hormones. The main one I want to talk about here is are the tyrosine derived hormones. These amine derived hormones these amine hormones are stored until they're secreted, and the receptor locations can either be on the surface or intracellular. But tyrosine is a parent amino acid for catecholamines and thyroid hormones. And the structures aren't terribly important, but you can see that they are derived from the tyrosine amino acid. There are also peptide and polypeptide hormones, and we'll talk a little bit about those with more of an emphasis on polypeptide hormones. These do not immediately enter the bloodstream. They are water soluble, so they don't pass through the membrane very easily or at all. So the receptors are located on the target cell on the surface of the cell. They can be stored in endocrine cell vesicles until needed. So let's talk about receptors and receptor-coupled signaling systems. Because these polypeptide hormones can't enter the target cell, they are termed first messengers, so they bind to external receptors. And then you have intracellular effects that are mediated through what are called secondary messengers. And these internal reactions, these are essentially the internal reactions in the cell. So these secondary messengers are low molecular weight signaling molecules such as cyclic AMP or calcium. They use transmembrane signal transduction cassettes to elicit their biological effects. We'll talk about more specifics of how this works. These Polypeptide hormones are part of a larger class of what's called receptor-coupled signaling systems. The most notable are these G-coupled receptors, which are all throughout our body. They're receptors for hormones, neurotransmitters, sensors, light photons, and other molecules. The main mechanism of how this works is the ligand itself, or the polypeptide hormone, known again as the first messenger, binds to the receptor, shown here, which activates a G protein in the membrane. In the second step, shown here, the G protein activates adenylate cyclase, which converts ATP to cyclic AMP, shown here, and so the cyclic AMP would be your second messenger. This second messenger continues down the cascade to activate protein kinases in the cytosol here. And then in the fourth step, phosphodiesterase inactivates cyclic AMP and turning that cell response off. Here's a list of the membrane receptors. The four main types are listed here. It's a bit of an eye chart. I don't expect you guys to memorize this, but it just gives you a sense of the different membrane receptors. And I'll talk about the four main ones that I want to cover here, notably the G-coupled protein receptors, ion channel receptors, these tyrosine kinase associated receptors, and then those that have intrinsic enzymatic activity. I'll bring this up a level here in a minute and show you what I'm talking about. So again, these four major types here are the G-coupled receptors, the ion channel receptors, tyrosine kinase link receptors, and then receptors with intrinsic enzymatic activity. Let's talk first about the G protein coupled receptors. These are a superfamily of structurally related receptors for hormones, neurotransmitters. As I mentioned, these are all over our body. They are integral membrane proteins. So they have these 7 transmembrane alpha helices, and then an extracellular N terminus and an intracellular C terminus, or an intracellular loop and C terminal end. There are 2 major subfamilies, small monomeric RAS-like G proteins and heterotrimeric G proteins. They have this molecular switch that regulates transmembrane signals from cell surface receptors to intracellular effectors such as adenylate cyclase, phospholipase C. So adenylate cyclase is the one I mentioned on the diagram there, but there are other intracellular effectors such as phospholipase C, phosphodiesterase, and ion channel effector systems. The effector specificity is conferred by the alpha subunit, which contains the GTP binding site and an intrinsic GTPase activity. What happens is the ligand binds to the G protein coupled receptor on the cell membrane by sitting in a pocket formed by the 7 transmembrane helices. So the G coupled receptor itself has no intrinsic catalytic domain. It recruits these G proteins, or the guanine nucleotide binding proteins, via this third cytoplasmic loop, which I showed on the previous diagram there, to couple signal transduction elements. Let's walk through the mechanism of how this works. In the inactive state, let me get my pointer right here. In the inactive state, the G proteins are heterotrimers with GDP bound to the alpha subunit, shown here. So here's our G-coupled receptor, and here's our G protein. The G protein is anchored to the membrane by lipid modification of the alpha and gamma subunit. So it's bound to the inside of the cell and the membrane here. The binding of the ligand to the G protein coupled receptor, shown here, again to this pocket formed by the transmembrane helices, drives exchange of GDP for GTP, shown here. This results in a conformational change in the alpha subunit leading to dissociation of the complex. So we have, down here we have dissociation of the complex. The activated G protein then interacts with one or more effectors to generate secondary messengers for downstream signaling, shown here. And then ultimately the signaling is terminated by the intrinsic GTP's activity of the alpha subunit. Some pathogens have exploited this, and an example is shown here. So cholera toxin from Vibrio cholera actually ADP ribosylates the G protein alpha subunit. So what this does is it prevents the hydrolysis of the G protein bound GTP, essentially resulting in a constitutively active form of the G protein. So then you get increased cyclic AMP levels within the intestinal epithelial cells, leading to phosphorylation of chloride channels. But essentially the downstream effects of this is you get large efflux of electrolytes and water into the gut, resulting in severe diarrhea. All through this mechanism of increasing the concentration of the secondary messenger cyclic AMP through ADP ribosylation of the G protein. Pertussis toxin is another example that has similar effects. So it enhances the cyclic AMP levels by two mechanisms. So it has the ADP ribosyltransferase, like the cholera toxin, but then it also secretes an extracellular and adenylate acyclase. Both of these result in cyclic AMP increase in concentration and inhibits neutrophil function, such as chemotaxis and oxidative burst. Here's a list of just a brief list of mammalian G proteins. This is just, I guess, for extra information here. I'm not expecting you guys to memorize this here, but it gives you a sense of some of the variety of G proteins. That's G protein coupled receptors. Let's also talk about ion channel receptors, with acetylcholine being the main example. What happens here is the ligand binding changes conformation of the receptor. So the ligand binds the receptor, and then a specific ion can flow through it. And then this ion movement alters electric potential across the plasma membrane. Some examples are neuron plasma membranes, which pass sodium and potassium, and muscle cells that use acetylcholine as the ligand. Botulinum toxin is a toxin that is a bacterial toxin that impacts ligand ion receptors. So in this case, acetylcholine is a neurotransmitter that's released by nerve cells that's at the neuromuscular junction. So this being your nerve cell, this being your muscle cell here. It releases acetylcholine from the nerve to the muscle to activate muscles. So the receptor is called the nicotinic acetylcholine receptor. What happens is the binding of the ligand, acetylcholine, is coupled to calcium release. The exact mechanism of this toxin is not terribly important, and there's actually different forms. But essentially what happens in all these different forms is the toxic fragment from the toxin prevents release of acetylcholine, this neurotransmitter, and cleaves proteins involved in the docking of neurotransmitter vesicles. But ultimately, you're disrupting this interaction between the nerve and the muscle, and thus leading to paralysis of the muscle. That's ion channel receptors. There are two more here, tyrosine kinase linked receptors being the next. This is a pretty simple receptor type. It has no intrinsic enzymatic activity, But what happens is the ligand binding results in formation of a receptor dimer, shown here, that activates tyrosine kinase that phosphorylates downstream targets. And then this phosphorylation can lead to downstream effects. This is in slight contrast with receptors with intrinsic enzymatic activity. So these are ligand-triggered protein kinases. They form this dimer upon ligand diamond, but the difference is the ligand receptor complex directly acts as a tyrosine kinase that phosphorylates downstream, that phosphorylates its target. The phosphorylation activates other protein kinases for downstream targets, and you got this cascade mechanism. But the important difference here again is it has its own intrinsic enzymatic activity. So here's a diagram of that here, but it's very similar to the previous one, except for the dimer itself has its own intrinsic activity. The last section here is I'm going to be talking about hormone-like lipids. These are important, and they are related to this lecture mainly because they are hormones or hormone-like in their activity. Some notable examples are arachidonic acid. This is a secondary messenger and a key inflammatory mediator. It's a precursor of eicosanoids, which are prostaglandins, prostacyclins, leukotrienes, and other molecules shown here. Again, the structures are important, but they act like hormones and signaled via these G protein coupled receptors. They have a wide variety of biological activities, such as modulating smooth muscle contraction, platelet aggregation, gastric acid secretion, salt and water imbalance. They also mediate pain and inflammatory response, which is obviously relevant to the medical biochemistry topic or this course. Let's talk more specifically about prostaglandins. So these, when you talk about conversion of arachidonic acid to prostaglandins, this involves cyclooxygenases. You guys have probably heard of COX-1 and COX-2. These are two different isoforms of the same enzyme in our body. COX-1 is expressed all the time, whereas COX-2 is made only in response to inflammatory mediators such as cytokines. These prostaglandins, as I mentioned, they stimulate inflammation, they can regulate blood flow, they can modulate synaptic transmission, and even induce sleep. The pain medication that we can buy over the counter is relevant here. They're these non-steroidal anti-inflammatory drugs such as ibuprofen and aspirin. They're actually cyclooxygenase inhibitors. So they reduce inflammation and fever by providing and provide pain relief by blocking this first step in the production of prostaglandins. Again, COX-1 is constitutively expressed. These are found in platelets, stomach, and kidneys, whereas COX-2 is more inducible. People sometimes dub COX-1 as producing good prostaglandins and COX-2 as producing bad prostaglandins because they are responsible for the synthesis of prostaglandins that induce inflammatory response. Aspirin irreversibly inactivates both of these forms. There are selective inhibitors of COX-2 that are treatment for rheumatoid arthritis, for example. Here's a chemical structures of some of these. Again, not terribly important, but just want to give you a flavor of what those look like. Okay, that's the information for today. This last slide here contains the study guide. I can run through this briefly here. I do want you guys to understand the general 6 steps of cell response and signaling. I had a slide on that. Just to understand that at a high level, how that works. What's the definition of a hormone, autocrine versus paracrine. I talked a lot about the 4 different hormone types, one of them being steroid hormones. It's important to know what they're derived from. What are catecholamine and thyroid what type of hormones are those? What amino acid are they derived from? And most of the lecture was revolved around polypeptide hormones. So I want you to know the 4 major classes that I talked about, and how do the cholera-toxins and pertussis toxins, which type are they relevant to, and how are their effects related to that type, the G-coupled receptor type. Which type is botulinum toxin relevant to, and which ions and ligands are involved there. And then the difference between the tyrosine-linked receptor versus the intrinsic enzymatic receptor. And then just the last 2 points, what is arachidonic acid, and how is that related to prostaglandins and the pain meds that I was just talking about. So that's it for today. Thank you for listening, and we'll see you next time.